Hi, everybody. Good to see you today. This is GIMBY Live, which is the Journal of Microbiology and Biology Education. This webinar is being hosted through the American Society for Microbiology. I'm Rachel Horak, and I work in the Education Department at the ASM. Today's speaker will be Caitlin Light, and we're going to hear today about training for responsible and ethical management of lab notebooks and cures, which are course-based undergraduate research experiences. So today, we're very delighted to have her talk about her recently published paper. GIMBY is an completely open access journal, all dedicated toward improving education in the K-12, undergraduate, and graduate spaces. So in a moment, when I bring Caitlin onto the stage, I'll post a link to her paper into the chat, and it is open access like all papers at GIMBY. She's going to give us a presentation for about 20 to 25 minutes. During that time, when you have questions, please put them in the Q&A box, and we will make time for lots and lots of participant questions at the end. Okay. So with that, if you have any questions, put them in the Q&A box. If you have difficulties, put them in the chat box directed toward me, Rachel. And with that, I'll turn it over to Caitlin. Thank you so much for being here today.
Awesome, thank you. Hi, everyone. Give me a moment here while I make sure I get my slides streaming for you all. All right, how's that? Everyone can see, hopefully.
This is a good screen share.
Okay, perfect. So as Rachel said, my name is Kate Light. I'm a research assistant professor at Binghamton University in our first year research immersion program, which I'll tell you a little bit about as well. I'm also joined by my former undergraduate research assistant, Sonia Hills, who's now busy working at her new position in Boston. So she's going to join us for the Q&A, and I'll take the lead on sharing our work from this paper. So Binghamton University, we have created back in 2015 a university-wide STEM program called the First Year Research Immersion Program. We enroll 300 first-year students every fall into this program. Like Rachel mentioned, it's a course-based undergraduate research program. And so the students are getting credit for courses while doing authentic research. I'll be calling that a cure from here on out. This program is very special, one of only a handful in the nation, where these students get to participate in 3 sequential cures, all within a specific discipline linked together, stepping them through the scientific process. At Binghamton, we have 10 different research streams or disciplines, and mine happens to be microbial biofilms in human health, so microbiology related, why I've found a home with ASM and GIMBY. This gives you a little bit too much detailed overview of our program. The first semester of our program is a research methods course, getting the students familiar with the content, exploring the literature, doing some meta-analyses and pre-proposals. In the second semester course, they get into the labs within their specific disciplines and start building more content knowledge, more skill knowledge, getting laboratory credits for here, and developing a research proposal for the research that they'll execute in our third and final semester, which is a really free-form, structured semester in which the students get to execute the research that they've been planning through the previous 2 semesters. Today, our talk's going to focus on how we implemented this activity in our second semester course, which is the first time these students are getting into the lab and learning some of these important scientific practices. The students that will be represented by our data here, we had in this study, we had 30 first-year students coming from majors like biology, chemistry, biochem. We have an integrative neuroscience program, so kind of across the board in biology, STEM-related fields. So how do we get to responsible and ethical conduct of research, which I'll start calling RECR so I don't stumble over my words too much. A little background history here. RECR training over the last few decades has become a big focus for national groups and for funding agencies, leading to sort of development of systems like city training to help get researchers more formally trained in responsible and ethical conduct of research. However, undergrads have sort of historically been left out of this increased push for RECR training, and just it's sort of been left to apprenticeship, one-off models. So this lack of attention to undergraduate RECR training is becoming more and more problematic as we see the push for these cure type classes across the higher ed curriculum. That led to the establishment of the Ethics Network for Course-Based Opportunities in Undergraduate Research, ENCORE. It was a program that I was lucky enough to be accepted into and mentored through to help sort of bridge this gap, figure out how we can utilize this increased flux of cure development to infuse RECR into our curricula. So the task for us as ENCORE fellows was to develop an RECR resource that could be used in cures, to try to field test that resource and share it. So luckily I made my way through all of those pieces over probably a period of 2 years with a lot of help from my mentors from that program, who I will thank at the end. But I first started out thinking, okay, well, RECR represents a really large area. What's a topic that's personally relevant for these students in my cures? Because we thought it was very important that it be something the students would care about. Otherwise, they're not going to have the motivation to really care about it and internalize it. So we definitely sat and thought about that for a while. But it pretty quickly came to the idea of lab notebooks, right? This is an important scientific practice that all researchers do on a daily basis. The infusion of authentic scientific practices is one of the 5 essential elements for creating a course-based undergraduate research experience. So that was very important. There's already RECR standards in place for lab notebooks. Of course, they vary by discipline, but there's guidelines to follow. This is something that undergraduates do on a daily basis in a cure, so it's something that's relevant and important to them. It's their primary responsibility. It's not something that they can pass the buck up onto as far as decision-making goes with RECR. They're the doers, and so they're in charge. So we felt like the relevance was really here for this. And personally, for me as an instructor, I've seen a lot of, and personally, and from my own experience, right, sloppy science is pretty easy to happen, and accumulation of those little errors and just little careless things can lead to somewhat irresponsible conduct of research, right? And it's really easy to do with lab notebooks. So Rachel, if you can help me here on this poll. I'm just curious what the audience, for those of you that are scientists, take a moment to reflect on how exactly you were taught, trained, or prepared to maintain a proper research notebook. Because this is sort of where we started. Okay, well, how did I learn how to do this thing? If I'm going to teach undergraduates how to do this, let me think how I learned. Give you guys a minute too. See if we get some answers coming in. You could also answer this question, how did you not learn? And bear with me on my voice today. It seems like it's quickly fading on me, but hopefully I can make it through. All right, how about 10 more seconds, Rachel, and then we can close it and just take a look at some of the answers.
You got it.
Thank you.
Okay, I'll share results.
Can't seem to see the results.
Okay, hang on. Hang tight. Let's see if I can get them. Okay, here, I'll read them to you.
Okay.
Okay, you ready?
Yep.
And they are anonymous. All right.
Perfect.
As an older grad student told me, and then I guessed and followed her.
Yep.
I learned as I worked, no one taught me.
Yep.
On the job, I haven't learned yet, actually.
Yeah.
A graduate student mentors during undergraduate independent research experiences. A graduate school course, a little during undergrad research directed by my professor.
Okay.
I was trained in a microbiology lab class and then in independent work in graduate school and my first research job after graduation. And looking at it, it looks like not a single person said that they learned during undergrad experiences.
Yeah. So that's perfect. Mixed bag there, but of course I'm going to pull out the sentiment that Sonia and I felt, and that was the on the job sort of just learn as you go, learn from people around you, stumble your way through it. Still trying to figure it out, right? For us, and especially I can speak for myself, that aha moment was like, oh yeah, I'm doing a great job. My lab notebook is so great. I'm so detail oriented. Then I went back to make my first poster, write my first paper, help my lab mate repeat their work, and where's this? Where's that? What did I do this day? What did I do that day? It wasn't until after I had to go back and use that notebook itself that I realized where the faults lied. And so that aha moment was something that we really wanted to recreate for the students before it was too late, before they had to go through what we went through and realize it when you needed that detail. And so I came across this idea of delayed gratification in a paper as we were putting this our own paper together. And it's sort of this idea that, you know, we get the instant gratification out of throwing our stuff in there and getting out of lab for the day and relaxing. And we don't realize the long-term pain that's coming down the road when we have to come back to the lab notebook. So we have to find ways to understand the value of putting in the time up front so that it's easier later. And to do that, we created this previous student lab notebook activity, which again, I'm going to keep using the abbreviation because that's a mouthful. PSNA is what we started calling it. I'll get into those details now. So Encore, as any good education research fellowship, guided us into creating student learning outcomes for our activities, and these were those associated with the PSNA. We wanted to teach students how to use lab notebooks in research, not just your chem inquiry lab reports, but a real authentic research lab notebook. How to assess how they were doing and revise them so that they were improving, and to understand and be able to predict the ethics around doing that or not doing that well. So I created this little timeline here so you could see what it looks like mapped to the semester. We did a pre-survey before right at the beginning of the semester. before the PSNA itself, we gave a little pre-lecture on lab notebooks, which I have a slide on in a minute. We did the previous students' lab notebook activity. Then we debriefed what went on with that and how maybe our ideas shifted throughout the semester. We kept working on notebooks, kept self-assessing with peer mentor and myself feedback, and then at the end of the semester, another survey. So our goals for this pre-activity lecture were just to share, like, what is a lab notebook, how do we keep one, why do we keep one, and get them a little bit of experience assessing lab notebooks. Specifically, we had them use the rubric that we created. You can find this in the appendix, I think it's appendix 4 of the supplemental material. We had them use this rubric on the PSNA itself, right? So just give them an idea of what to do once they got to the lab. One thing I want to point out about our rubric, we really tried to gear the quality categories towards the effects of the quality on the downstream user, right? From a scale of 3 to a 1 or 0, 3 being any outside researcher could pick up your lab notebook, understand what was going on, and replicate the experiment. And 1 to 0 being that moment of frustration when you can't even repeat your own work from your own notebook, right? Because that's sort of something easy to grasp. Okay, like, I can't even repeat this, or yes, I can give this to my colleague in another stream and they can do this work. This is only one piece of the rubric, so I encourage you to check that out if you feel like that might be useful in your course. The previous student's lab notebook entry. Here's the meat and potatoes of the activity itself. But what we did was we created our own student lab notebook entry. And we purposely, Sonia and I worked pretty hard to build in some pitfalls to this lab notebook that would prevent the students from readily understanding why the experiment was happening, how it was happening, where to find reagents, how to use them, how to use equipment, where to find equipment. And the students were asked to, just like many of us have done in grad school, oh, you know, Joe did that back when he was here as a grad student, grab his notebook, you should be able to repeat his work just fine, right? And then we're banging the notebook on our head. Okay, I can't quite figure this out. That aha moment for us, we're trying to create it here for students. So our activity was centered around a bacterial growth curve, which is a pretty simple entry-level assay that we would use in a microbiology lab. It fell early in our semester training and semester research project. It's easy enough to grab onto and try to do. So we gave this notebook to our students, and they were asked to basically come to lab and try to execute the experiment from this lab notebook, right? And we'll show you some of the cool results as we go. We created a guide in the appendices as well for you to adapt our activity to your own assay technique or what have you in the future as well. Or you can plug and play with ours if you would like. And then in the post-activity lecture, we just sort of discussed and debriefed, right? Like, what was frustrating? Well, obviously I didn't probe them, prime them with that, but how did you feel during this activity? What were some of the challenges? What did you learn, right? And, you know, a lot of the sentiments were frustration. I'm sort of like keying you in on some of the results there. But then we looked at that previous student's lab notebook entry again, and we reassessed it with the rubric and talked about how we would revise it to improve it, right? And unpacked a little bit more like, okay, what are the consequences How do we predict what's going to happen if we aren't good at maintaining this professional standard? Encore help assessment, right? You create this great activity. Does it work, right? Does it meet those learning outcomes that we set out to do in the beginning? So here are our results. This graph that you can see here is showing sections of our laboratory notebook across the x-axis. And on the y-axis, you can see scores ranging from 0 to 3, as you saw with the rubric earlier. In gray, we have instructor scores of that specific student's notebook for the PSNA. Myself and a couple other instructors within our FRI program scored this PSNA. Black was the student's score in the pre-lecture activity. What did they think about this notebook prior to having to use it? And white was the student's scores after they had used it in the lab in our post-activity discussion, right? And what's really cool here, you can see in almost every category, the students were higher than us as instructors prior to the activity. They thought it looked really good. It was organized, it was pretty, there looked like there was a lot of detail. But once they had to use it, their scores are coming down and they're a bit more in alignment with how the instructors are scoring them. And specifically the two areas of the lab notebook that we put the most pitfalls in, the background and the protocol, we saw significant differences between students' evaluations of this notebook before and after. And overall, when we add up the total score on the notebook, there was a statistically significant difference on the entire score. The students gave the notebook entry at 84 before they had used it, and it came down to a 74 after they had used it. So that's pretty cool. Here was just a quote and a few words that were mentioned as we did this debrief. They understand how confusing it was, and they don't want others to have that same confusion, frustration, stress, and anxiety, right? All those same things that we felt after we had realized how bad we had done with our notebooks, right? Despite our efforts. So we felt like we created that moment for them, and it was there to act on, and if the importance was there for them. This graph shows my students, their scores on their own notebooks over this second semester course. So they submitted 14 different notebook entries, and you can see this slow but steady climb in their scores from the beginning until the end. When we tested the difference between the score in the beginning, entry 1 and entry 14, it was statistically different, this growth pattern. And we think that taken together, these results sort of show that the activity and the repeated use and evaluation and feedback of the notebooks over the course of the semester are actually achieving our goal in teaching students how to maintain a quality notebook, right? These data are our survey results, and I have to sadly point out we have an erratum associated with our paper here, which I encourage you to check out. Otherwise, some of the results might be a little confusing. But one thing we did during the analysis was to reverse code some of our data so that they were all in the same direction, as in the correct answer or confident answer is towards the green side of the graph. And when we did that, we did not change the coding on our question. So I encourage you to check that out. The data you're seeing here is from the erratum. Not a lot of changes, and this is something that we're interested in exploring further. We think that students come in with actually a pretty good understanding of what ethical and responsible conduct should be. And the problem comes in with when they are responsible for that conduct and how to apply it, right? And hopefully I can provide you some evidence of that here as well. So these were two of the questions with the most change. Because this is a cure, you know, it's a course, the lab notebook's not my property and I can't take it with me. We saw a good shift in that, which was great, right? They learned what the true purpose of a lab notebook is. And that speaks to the second question here as well. It's not just for my instructor to grade me, it's to help others repeat my work and to document my work. So we thought that those were some really cool findings. We had 2 case studies on the survey as well. This first one is about data falsification and fabrication in the notebook. They knew right and wrong, right? Pretty much, we're not really clear why we had one shift back in the other direction, but they knew right from wrong. You can't do that. But when we asked them a question about how to handle repeated experiments in the notebook in this class, that's where there was a lot more confusion, right? Can I just copy and say, oh, check out page 6, or do I need to record X, Y, Z particular details? And we saw a great shift in this response from before they had started working with notebooks and going over the activity to the end of the semester. So that was really great. Some of my favorite data are found here. These are quotes from our programmatic end of semester reflection essays. So this is something that we collect as an entire FRI program and wasn't originally related to our study for the PSNA. All we do for this is ask students in 300 words to reflect on their personal and professional growth as a response, as a result of this semester, right? And so we were like, let's check them out. Let's see if there's anything in there, right? We've been working on lab notebooks a lot. We've had a lot of activities. We've been revising, improving, etc. Let's see what they talked about, right? Remember, all we asked them to do was, how do you feel you've grown personally and professionally? 12 out of the 30 essays mentioned lab notebooks or data management as they sort of tangled together. We were so excited by that. You know, of all the things they could talk about, and we focus on a lot of other things in FRI, they keyed in on this, right? While it seemed tedious, in retrospect, it was really important, and it helped me perform experiments that other people can replicate, right? I've gained a much deeper appreciation for the value of writing down protocols. And oh yeah, I've painfully learned how hard it is and why it's so important, right? They're speaking to that value and that importance. And that was so exciting for us to see that in an unprompted response. Long time after the PSNA activity itself. So sort of the conclusions that we took away from these data, we think that the PSNA and the iterative practice and feedback using the rubric throughout the semester on their notebooks may contribute to improved lab notebook abilities. We see a significant change in their understanding of lab notebooks. So if we, I forgot to mention, if we sum up all that survey data and we look at the score from pre to post, there's a significant change towards the positive or correct or confident side of the Likert scale from post. So we are affecting a change in their understanding or attitudes, depending on the question. We are also thinking that students understand what's right and wrong for a hypothetical person, but they're less clear on when or how that applies to themselves, specifically in a cure setting, this sort of hybrid between a course and a research experience. It's kind of gray there for them. We see a disconnect between knowledge and application of RECR in the cure setting. And we think that we can see that students are reporting an understanding on the value of lab notebooks in research and in cures. So in the future, we would like to consider tracking and assessing this skill beyond our second semester course into our third semester course of FRI, and even looking at other research streams in our FRI program or other cure courses that we have running at Binghamton. Other future directions, more broadly for RECR and cures, I'm really interested in exploring this idea of like, why do these students seem to know what's right and wrong? I mean, this is just human nature, perhaps, right? But they can't apply it or put it into action, or they don't know when or how. So exploring that and continuing to help figure out how we could keep infusing meaningful RECR into cures as we see more and more research experiences pop up into our undergraduate STEM curriculum. So with that, I'd like to thank a few people. My undergraduate researcher, Sonia, for many hours in developing, assessing, and writing up this activity. The FRI program, my director, Megan Fagley, and my other research educators who helped me field test this instrument, the students from my cohort who engaged in the activity, a previous undergrad, Emily Rail, who helped implement and field test this activity. And then last but not least, the Encore Network for providing the mentorship, the teaching, funding, and time and structure to explore this really important idea. I could not have done that without all of these people. So thank you so much. And I hope you have some great questions that Sonia and I can share with you.
Thank you so much for telling us about your paper and your findings. Sonia, would you like to join us? And you can stop sharing now, unless something comes up in the questions that you want to refer to a figure, you can bring your presentation back on. Okay. So we have lots of compliments. People have really enjoyed learning through this. I have a couple of questions for you, and I'll just throw them out there, and if either one of you want to take them, you may. Sonia, if you wish to turn on your camera, you may too. All right. So first question, it was interesting to learn how this activity helps students realize that noting the why in the background and how in the protocol of the experiment is a crucial part of a lab notebook so that the experiments are able to be reproduced. Question, did you also design activities around the later part of record keeping? For example, data analytics and results, which incorporate concepts like data normalization or statistics or image modifications. Perhaps you did this later in the semester. And they're asking because the category doesn't appear to show much learning in figure 1, but is an important part about data replication.
Yeah, definitely. That's a great question. Sonia and I did build issues and pitfalls into all of the sections of the PSNA. The way that our assessment around the use of that activity in the lab without help, the way that happened, the students hadn't had time yet to have to write up their entire notebook entry for that experiment. So that's something we probably would like to explore in future work as well, is having something separate to unpack that. The way we sort of handled it as far as teaching and mentoring them was to give feedback with the rubric on their notebooks and work them through the process of like why this notebook was good, why this one was, what was missing, what we need to do in the future. And we think that we can say that we're making an impact on that as we see those scores climbing across the semester. But there was nothing intentional about those later sections with regard to the aha moment. That was definitely more geared toward the background and the intro.
So the next question is, thank you for the insightful talk and discussing this impactful activity to teach students about good record keeping. As we saw in the polls, many people that are current scientists and educators didn't learn these skills as part of their integral part of their undergraduate training. The people, when we polled, people got training as undergraduate independent researchers, but not outside of the coursework. Okay. So my figure is regarding the data in figure 3. My question is regarding figure 3. The results from the pre and post assignment student survey highlighted one category with the most change. And you had a slide about this. My lab notebook is my property and I can take it with me. It's interesting. It speaks to the accountability that a student feels toward their mentor or lab versus themselves. Do you believe this realization alone contributed significantly to reduced sloppiness in record keeping by the students? What are your thoughts? And maybe Sonia, maybe since you're, maybe you can comment on this as well.
Sure, I'd be happy to. I mean, I think it definitely plays a part in it as students realize that they have a responsibility greater than themselves to keep a responsible lab notebook. And I think also the fact that they are working in the Cure, they work on a team. So I think it may start to key in throughout the semester that if they're working on a team, their lab notebooks need to be useful to their team members as well. And they can't complete their overall project just relying on themselves.
Yeah, that's definitely an important piece of context that I left out for sure. So it's sort of forcing them along the way to make sure that the notebook is good enough for a downstream user, even if that downstream user is their like current and immediate teammate. Yeah.
Thank you. Next question. The pre and post assignment survey asked a very important question regarding how seriously a student should take the activity and maintain lab notebooks, not just for the sole purpose of grading. What can we do as educators to convey this effectively to all of our students, maybe not just in a Cure, so that they realize the larger ethical context here?
Yeah, that's a tough one, right? And that's really how we landed on this activity, right? It was for us. How did we eventually realize the importance? I think that Sonia and I are both type A, highly detailed people. And like we had always thought that we were maintaining great, like there's so much detail, right? But detail doesn't always translate. We weren't really able to grasp the importance of it until we needed it ourselves later. So when thinking about how you might apply this to a non-research related course, or, you know, let's just say to students who aren't heading down a research pathway, I think you need to identify what is that thing that's going to be important to them later to find. This could even be structured with data management. How are you saving your files? How are you labeling them? How are you storing them? Where are you putting them? I could see where you could create something like this for med students, right? But I think the most powerful thing about this is creating that personal moment. Like, if I don't do a good job on this, I can't do my research in the third semester because I won't be able to remember how to do it, right? And in our third semester, we tell students all the time, they'll come ask us questions and we say, I'm pretty sure we did that 2 or 3 times in semester 2. Don't you have a notebook for that? And yeah, and 9 out of 10 times they are able to figure it out, which is great, right? And then sometimes we just have those not so helpful lab notebooks, and thankfully we have our lab mates to help on that. But that's a tough question.
Maybe for the next person to study.
Yeah.
Now, seeing that it was a cure, and do you have one lab notebook for a group of individuals, or did each student have their own? And follow up is, what are you using for their lab notebooks? Are you using the marble composition books or blue books or something else?
You could take this one, Sonia.
Okay, yeah, I can answer this one. So it varies between the 2 semesters. So in semester 2, which is where we implemented this activity, students are doing a lab notebook in pairs. So they also are doing their experiment together in the lab and then also write it up as a pair. So they have that reliance on somebody else at that point and are able to help each other. And then once they take that skill to the 3rd semester, they're working with their entire research team, which is about 5 or 6 people. And we actually implemented this during a peak COVID semester. So it was entirely an electronic lab notebook using Microsoft OneNote. So the students were in the wet lab and like actually doing the pipetting and lab work, but their formal write-ups were in an electronic notebook.
Yeah, and I can I can plug another paper there that we published in GIMB right around the same time, I think it was last spring, on how we handled remote cures during COVID for our like entire program. I'll see if I can find that and maybe pop that in the chat for everybody.
And Sonia, what's your thoughts? Do you think that the same result, the same data that you found, do you think that it would hold up if you use a paper notebook as opposed to an electronic notebook? That's a completely hypothetical.
Actually, it's not. When I was in a cure, so when I went through this entire cure sequence 2 years before this activity was implemented, and back then there was, Dr. Light did like a very mini version of this where she actually handed us a bound composition book from a previous student, and we had to do the same. It wasn't as formalized, and it was more like a 20-minute activity overall with no lectures or surveys, but this happened with the composition book, and I don't think it had the same effect. I think we understood some of the things, especially the pitfalls that were in that particular student's notebook, but they weren't intentionally built in like that. So it was still missing a lot of the key things that we needed to know about. But I think the iteration and formal grading with the rubric of it was, I think, really important and didn't carry in the composition part. But I think it also doesn't, I don't think the electronic part of that necessarily made a huge difference there.
When I talk to instructors, they're always trying to make that decision. Do we have hard copies printed, or a lot of instructors are trying to use electronic notebooks. And the people I talked to last week, they were like, well, we tried electronic lab notebooks, and students didn't prefer them. They preferred writing it out in a paper notebook. So
yeah, and we still see that with our students, and we sort of are still working with them to figure out what that hybrid model looks like. A lot of them have a composition book and take their notes and observations in it, and they just get transferred to we call it an ELN, electronic lab notebook. They get transferred to the ELN later. And then we also started encouraging, because this is a microbiology lab, we don't have the laptops out where we are working with our cultures. We have a clean space for them. So when they needed to use the lab notebooks, they were trying to just remember it. And I mentioned that earlier, them asking us questions. We're like, that's in your notebook. So we started to encourage them to print copies of their ELN. And we talked about that being a twofold decision, the decision of having it in the lab, which is helpful, but also of proper RECR, backing up and having hard copies that we sign and document and can't change anymore, right? Because when it's electronic, especially with a free platform like OneNote, it could be a bit easier to go back and change things. So we're teaching them to export to PDF, save those and print them. So there's hard unchanged copies as archive as well.
I'd like to add on to my previous response too. And I think one element of the electronic notebook that I think may have helped students learn and improve with this activity is the fact that it is editable, and the fact that you can sort of go in and add things and sort of like review in a way that you can't on a paper notebook. And also, I think one thing I really struggled with the paper notebook was the fact that I just couldn't write as fast as we were moving. And so just being able to jot down things on the paper notebook, but then later being able to really formalize it with that time to sit with it, I think is a really big benefit to the electronic notebooks.
Thank you. All right, I have another question. You gave the students a single version of omissions. What challenges do you see if instead of giving a single version of omissions to the class, what if you gave multiple versions of omissions to different groups in the class and have the students discuss their experience?
Yeah, that's a fabulous idea. I'm not sure, Sonia, if that's something that we had discussed or some feedback I got from Encore as well. I love it because I know there's another team building activity like this where, students are supposed to create like a craft project, and you give one team scissors and another team glue, and so they don't all have the same stuff and they get stuck at different points. So we thought that might be a fabulous idea. It's a lot more intricate and probably hard to manage, and also keeping these new freshman students safe in a microbiology lab. So we started simple, but I think that's a really cool idea, especially if it's paired with a discussion and a sharing and like, let's revise this and create a notebook together from the good and bad bits that we all had.
One participant wrote in a nice comment that one thing they've tried is having one student be the scribe each week when the educator has them work in groups. And that scribe does all the writing either on a laptop or a paper notebook, and everybody shares their notes. So really getting at the team aspect, and it's good to definitely share those duties.
Yeah, and that's exactly the essence of the paper that I shared. So in COVID, we had to cut the lab. That was the year that we did this activity, right, Sonia? Was when they were on the iPods. Yeah, so we had to cut in half, and the way we kept the Zoom partner engaged was the in-lab person doing the physical, technical lab stuff wore a harness with a completely protected lab-specific iPod that they Zoomed on, and the person who was virtual on Zoom was in charge of the notebook for the day, scribing as they were going along, so that it wasn't quite as boring in Zoom land. You had a really important purpose to sort of try to keep up with what was going on instead of just passively watching. So I love that strategy as well. That's great. And switching roles, and yeah.
So it looks like we have no more questions. So at this time, I'll remind folks that we have another Jimby Live coming up December 2nd. It's a Friday at 1 p.m. Eastern. Again, it'll be free for anybody. It's going to be called Microbiology Labs with virtual and hands-on parts. and how it promoted student success during the pandemic. And it's not, you know, we're all face to face with many institutions have fewer COVID restrictions right now. But I think that we can really learn from some of the papers that came out of how educators adapted to the COVID pandemic, about how we can continue to improve student learning so that every single student can learn, no matter what its consequences. Okay. And then, so that's free. And of course, all Jim B Lives are recorded. And into the chat box, I just put the link to view any previous Jim B Live episode. And so there's our YouTube playlist. And the recording of today's episode will be available sometime next week on our YouTube playlist. So you might want to go in and bookmark that page. And oh, hang on, I didn't send it to everybody. I sent it to the wrong people. Okay, so there you go. And you should have it. So there you go. The YouTube Jim B Live playlist and the link to go register yourself for the December 2nd are now in the chat. So thank you so much for joining us today. Thank you to our speakers and to our authors. And we hope to see you at a future event.